in progress.
Good morning.
Morning. Are you driving?
Morning.
Any electricity at my house, so I'm using my phone. But
You have no electricity at your house?
right, we had an ice storm down here last night. I don't know if you guys did too.
It was more sleet and rain, but it wasn't ice.
You can hear me.
Oh, that really stinks.
Good morning. Good morning, Professor.
Good morning. All right. First, I know I got an email from somebody this morning, but outside of that, any questions or concerns? We're gonna try to finish chapter 8, the heart today, chapter 19. One thing that I should mention that I didn't mention the last time was with me here.
Sorry, I think I lost you there.
It's all right. I understand.
I might be spotty today, just so you know.
I got it, and I understand. Okay, so there was, coming back really quickly here, one thing that we did not talk about is, if you remember from 121, muscle can generate, especially skeletal muscle, can generate ATP anaerobically when it needs to. Meaning that it doesn't have to have oxygen present. It can generate it. It can generate it via fermentation or glycolysis rather than using aerobic respiration. Cardiac muscle can't do that. It does require oxygen, and it can only make ATP via oxygen aerobically. And the result is that we have hemoglobin here, but we also have myoglobin. Myoglobin meaning a derivative of hemoglobin. Bear with me here. A derivative of hemoglobin. Myoglobin. that specifically holds and stores oxygen, derivative of hemoglobin, that stores and holds oxygen. The last chapter that we covered in 121 talked about how muscle generates ATP anaerobically, and it bear with me, it has an oxygen debt repayment plan that it can use when it starts to run out of ATP. That doesn't work here. It always has to be able to have plenty of oxygen stores available. Therefore, it has a lot of myoglobin available. It has a lot of glycogen available all the time. Because it doesn't do fermentation. It's not going to generate lactic acid. And it can only generate ATP via aerobically with oxygen. I realized as I was looking through a list of objectives that I made that I needed to cover in this chapter that I hadn't talked about that when I talked about muscle. But keep that in mind that this muscle, specifically heart muscle, does not generate ATP anaerobically. It always is aerobic, meaning it has to have oxygen. And in order to do that, it's got to have stores of oxygen holding. I mean, it has hemoglobin running through it, but it also has myoglobin, which is specifically storing it in oxygen in muscle. And it has glycogen, and it will not generate ATP anaerobically. So therefore it has these extra stores available for its use. Questions about that? Mom? Abby, out of there. Abby, out of there. Sorry, my cat's going into my mom's room and she does not like that. Abby. So remember the muscle tends to flow. It has intercalated discs. Therefore, it has specialized gap junctions that allow ions to flow from one muscle fiber to the next, allowing muscle to act as one functional syncytium. I think it's syncytium. Allowing it to contract and relax all at the same time. Another thing about the heart that makes it very unique is it has an autorhythmicity, meaning it's going to contract rhythmically as a result of action potentials that it generates by itself. The nervous system will influence this rhythm. We'll talk about that towards the end of this PowerPoint. But but the heart on its own can generate a rhythm, and it generates a series of action potentials all on its own via its autorhythmic cells. which initiate and conduct action potentials that are responsible for the contraction of working cells. Therefore, it's going to generate action potentials, so electrical pulse that will trigger the contraction of working cells or cardiomyocytes. I had an interesting experience with this, and if it hadn't have been 4 a.m, I would have tried to get a picture of this. But during my second master's program, I was working with baby chicks, and they were really, really young baby chicks. They were more chick embryos, and they were about, for human babies, they probably would have been about 3 weeks, 3 to 4 weeks gestation, not very far along. And what I did is I was studying limb development and kidney development, and how drugs interacted with and altered that. And with that, what I would do is I would open these embryos up, these eggs up before, we're really early in development, and treat them with something. And then close them up for a couple days, and then come back and fix them later, cut them out of the egg and fix them. And there was a time where I was doing it, and I'd had a death in my family. That's why it was 4 a.m. I'd had a death in my family, and I was flying from Idaho to, I'm trying to remember which death it was. I think I was flying from Idaho to Arizona. And I was in the lab, and it was really, really early because I had to get this done before I could leave on the plane. And I had my car was all packed, mind you. And so, 4 a.m. at the university, trying to get this done. It was probably closer to 2 or 3, but really, really early. And I'm sitting there, I'm trying to clean off these embryos. I'm cutting them out of the egg so they don't have any nervous system stimulation, no extra push to make them be alive, because I've cut off, I'm essentially just, they grow on a membrane on the top of the egg. And then you cut them out and you fix the membrane. And so I've cut out this little ring on a membrane. Bear with me, I'm going to see if I can find you one. I'm trying to discard this, and it's not going to work very well. So they're about this big. Here's the head. Here's the heart. Here's about where the developing wings are going to form, and the developing feet. And they were actually they were closer to this stage, not that guy, closer to this stage. So really, 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 really early in development. And so I cut them out, and at that point, they're clear little, they're on a clear membrane. By the time that I would cut them out, they're on a clear membrane, and I'm cleaning them up, And so what I'm doing is there's an extra membrane on top, and I'm trying to remove that extra membrane. And the way you do it is you kind of clear it off with a piece of forceps, and then you clear it off with a piece of forceps, wash them a little bit in saline solution, and cut just as much as you can away from the body, and then stick him in the fixative. And as I'm doing this, he doesn't, this embryo, because, once you start cutting this all away, they bleed out. There's not a lot of blood in there. But once you start cutting all this away, they bleed out. So there's no real blood here either. Once I'm to the point where I'm almost got him completely cleaned up, there's no blood in there either, mind you. And after I've done this, then I'm cutting off whatever I can before I stick him in the fixative. And I look down, and his heart is beating against the plexiglass of the petri dish. And it was the only thing that could be beating at that point. And it was, so it was adhered to the plexiglass. Nope. He was adhered to the plexiglass. Here's the plexiglass, and he's doing this along the top of the plate. And I, if it hadn't have been 3 a.m, and if I hadn't have been trying to make it to get to my flight on time, I probably would have been more like, oh, I probably would have tried to video record it or something, rather than just saying, oh, that's really cool. But what it showed was that the heart by itself generates a rhythm. And it will for a while after death. And so we're going to talk about how that rhythm's generated right now. I have another picture of them. So these autorhythmic cells are going to generate action potentials that are responsible for the contraction of the muscle tissue of the heart. And the other 99% of the heart is cardiac muscle, which is actually going to do the mechanical work of contracting and pumping the blood. So we have the autorhythmic cells, and remember talking about action potentials in 121. These action potentials work very, very similarly. Remember, you have a membrane potential, and there's depolarization, and then depolarization results in reaching threshold. And then you get a rapid depolarization and then a repolarization. Well, it's very similar here, except there's no resting membrane potential. If you remember from 121, and I'm finding another picture of it. There's a resting membrane potential. Actually, let's do this one. Resting membrane potential, where a stimulus is applied and then it moves up. So this is the action potential in a neuron. Slowly it moves up, and if it reaches threshold, then you get a rapid depolarization because you open those voltage gated channels. Coming back to you. Can't see any faces right now, so coming back. Well, in the case of the autorhythmic cells of the heart, there's no resting membrane potential. So it's not stable. There's no flat line and then having it go up. It's a rhythm. The whole time it's a rhythm. Because there's a slow depolarization that's going to be occurring because of the opening of what we call funny sodium channels. They're just kind of weird little channels that open up and allow sodium in. And you remember this, you remember that's how it worked in the action potentials of the neurons, that we had sodium that came into the cell. So it starts that way in the heart. You have these sodium channels that start to open up. And there's a rhythm here. Remember, this doesn't require any nervous system input. It's a rhythm that's consistent. So slow depolarization due to the opening of funny sodium channels. And then as you reach a certain level of depolarization of the membrane, calcium channels are going to open. And they're going to allow calcium in. In addition to the sodium, now you're allowing calcium in. And once you reach threshold of -40, there's a rapid depolarization by the opening of voltage gated, more rapid gate voltage calcium channels. And this leads to a rapid depolarization. So it's not sodium in this case that's leading to this rapid depolarization like it was in neurons. Now it's calcium. That's a key point to remember here. It's calcium now, it's not sodium. There is help of sodium here, but calcium is the key player. Once the membrane potential reaches about 0, then the calcium channel starts to close and potassium gates are opening. And we get the rapid repolarization. And with that massive potassium repolarization, we then are back to our more negative membrane potential. And then it starts all over again with the funny sodium channels opening and then the calcium channels opening. And there's a video that I want to watch here really quickly. I'm trying to remember just how long it is, and I can't remember. So I put it in here last week, and I can't remember how long it was. So let me just see. there's two videos, I think, and I can't remember if this was the one with the guy that discussed the capillary exchange. It was a longer one. But he's,
I have to stop and tell you.
yep, it is him. Let's see how long it is. I think it's worth it. We'll watch this one.
Hi everybody, Dr. Mike here. In this video, we're going to take a look at how the SA node, which is the sinoatrial node, and the AV node, which is the atrioventricular node.
Actually, I'm going to wait on this because I couldn't remember where he started, but I don't want to go. I want to, before he gets to that point, want to move a little further along.
How they send their signal, their electrical signal.
Bear with me. I apologize. I'm realizing I don't like where he's starting because we haven't talked about the SA node and the AV node. Do you remember talking about the SA node and the AV node? We talked about it briefly when we talked about the heart in lab. Somebody,
Yeah,
you remember what was it about?
About the SA node and the AV node. It's the conduction system of the heart, the electrical conduction system of the heart. Right?
Yes.
Okay, sorry.
You're right. You're right. Let me go through this question really quick. Pacemaker potential is a result of what?
was it sodium?
It's actually, it's calcium. Remember, in this case, it's actually going to.
Oh, yes, calcium.
So the major difference here between the two is sodium starts it between this type of action potential and the nervous system action potential is that calcium starts it. No, sorry, sodium starts it, but calcium actually results in the rapid depolarization. That's the major difference here. Calcium didn't really play a part last time, as far as I can remember. If it was, it wasn't significant enough to talk about. However, here, calcium is the major player in the depolarization. Sodium starts it, but calcium doesn't. Does that make sense? It's the same. I mean, it's the only difference between it and sodium is that it has a 2 positive charge. Its concentration is still greater outside of the cell, just like sodium. So it's the one that's the key player in the depolarization here.
So it's E, yeah.
Yeah, it's E. So how does the action potential spread? How does this electrical pacemaker action potential spread? The pacemaker is the SA node. So the SA node, which is the sinoatrial node, initiates this rhythm of action potentials. And the excitation, then the signal of current, just like your electrical wires at home, the signal of current then spreads throughout the adjacent atrial myocardium. And it spreads through starting in the right atrium, moving into the left atrium, down the edges until it reaches the AV node, the atrioventricular node. But it actually slows down a little bit because the muscle here, or the autorhythmic cells don't have a smaller density of gap junctions. And there's also a lot of connective tissue here. So smaller number of gap junctions, a lot more of the fibrous skeleton of the heart. And it slows it down, which is going to be important later. It slows it down, but then the action potential spreads through the AV node into the AV bundle, the atrioventricular bundle, down these bundle branches of His. It goes down to the apex of the heart, and then up through the Purkinje fibers, distributing an excitation throughout the ventricular myocardium. He's in
here. He's okay.
Okay. These subendocardial conducting network, these are what we call Purkinje fibers. I'm going to put that on another sheet page. So the subendocardial conducting network are the final branches or final extending branches of the AV bundle. They are called the Purkinje. Another word that we call them is the Purkinje fibers. Now that I've explained that a little bit and we've kind of reviewed that, let's look at what this video says.
I have to stop and tell you guys about Lecturio L.
Hi everybody, Dr. Mike here. In this video, we're going to take a look at how the SA node, which is the sinoatrial node, and the AV node, which is the atrioventricular node, how they send their signal, their electrical signal that we term an action potential, to the rest of the muscle of the heart to tell it to contract. So what we need to go through is, firstly,
So these are the autorhythmic cells, meaning they're going to generate the rhythm. And they're going to send a signal to the cardiomyocytes. Cardio meaning heart, myo meaning muscle.
if I were to draw the heart up itself, and just very quickly, I've got the heart here. We know that there's a fibrous bit of tissue here that separates the atria, the two chambers at the top, and the ventricles, the two chambers at the bottom. And we know that there is a specialized type of myocardium, which is heart muscle cell, a very specialized type that sits at the back of the right atrium that we term the SA node. Now, these cells spontaneously depolarize. What's that mean? It means they spontaneously send an electrical signal. They actually do this around about 70 to 100 times a minute. And when they send this signal, they spread the electrical signal to the surrounding heart muscle tissue in a fashion like that. Now, as it spreads that signal through what we term the myocardium, the heart muscle of the atria, That then tells the heart muscle to contract, and they squeeze and push the blood down into the ventricles. Now, the electrical signal wants to spread down into the ventricles, but it can't because of this fibrous bit of tissue here. But the electrical signal can bottleneck itself
So
through another.
that's what forces it to, because of that fibrous skeleton that's blocking it from moving down around the sides of the heart. That's what forces it to go through the AV node. If it had a choice, it wouldn't try to go through the AV node. It would maybe go around the sides, or it would go through all three. It would take all three pathways. But rather than taking all three pathways because of the fiber skeleton, it has to move this way and go down the atrioventricular septum.
Specialized group of myocardium that we now term the AV node, the atrioventricular node. And the action potentials are being pushed through this AV node, and therefore the heart muscle doesn't contract for 0.1 of a second. There's a break between the atrial contraction.
So I told you there was a gap. There's a slowness when it spreads from the AV node into the atrioventricular bundle. That's what he's talking about there.
And the ventricular contraction of 0.1 of a second. This gives the heart enough time for the ventricles to fill with blood after the atria contract.
Did you hear what the significance of that was? Why there's that slowness? Because it gives the ventricles a chance to fill with blood. Remember that blood is flowing like a fluid. It's flowing like water. Maybe a little bit more viscous, but not significantly. I mean, I've watched enough NCIS to see how blood tends to spill out of somebody. It's not that viscous. So it's flowing like a water. And so that gap, that little moment where the signal pauses and slows down as it moves into the atrioventricular bundle, gives the ventricles a little bit more time to fill up with blood. That's going to be important in a little while here. It's going to be really important in a little while here.
Once that 0.1 of a second has occurred and the action potential spreads down the AV node, it goes down into what we call the bundle of His, and that spreads to the left and right bundle branches, and then they send electrical signals through the ventricular heart muscle, and as it spreads through, that action potential then tells the muscle to contract and blood gets squirted out. If it's the right ventricle, it goes to the lungs. If it's the left ventricle, it goes to the body. So what we're talking about is today the SA node and the AV node, how do they send their electrical signals? If I said the AV nodes spontaneously depolarize, they just basically stimulate this action potential all by itself. How does it do it? Well, let's first have a look here. Actually, let's have a look here at a cell of the SA node. So I said there's specialized myocardium, so it looks a little bit different here compared to normal myocardium, but it is connected. You can see there's a conversation that can be had between the SA node.
There's your gap junctions. It's one of the gap junctions, one type of gap junctions.
And the myocardium. Now, the first thing you need to be aware of is that all cells of the body have a very special pump called the sodium potassium ATPase pump.
You remember that the sodium from last semester, the sodium potassium ATPase pump, which is going to maintain that resting membrane potential. Remember that it keeps the charge allows you to keep the sodium concentration higher on the outside of the cell than the inside of the cell. It's also, and it keeps the potassium higher on the inside of the cell than outside of the cell.
Let's draw this sodium potassium ATPase pump right here. And what it does is it uses ATP. We know ATP is the energy currency of the body. Uses ATP to throw 3 sodium out of the cell and throw 2 potassium into the cell. Now I want you to think about that. If we throw 3 positive sodium out of the cell and throw 2 potassium into the cell, where do we have a net positive charge? Well, we've got 3 positive things outside, 2 positive things inside. The net positive charge is outside, which means the net negative charge is inside. And if you were to compare those charge differences, what you would find is inside compared to outside is negative 5 millivolts, okay? So on this table here, we've got 0 millivolts down to negative 90 millivolts up to positive 20. Negative 5 millivolts is only around about here. Now, this isn't all that's happening. But what is happening due to this is we're accumulating a whole bunch of sodium outside the cell, and we're accumulating a whole bunch of potassium in the cell. That's the first thing. Second thing is this. This cell is quite...
Zena, for you who's taking 121 at the same time, this is what we were starting to talk about the other night.
Yeah, it makes sense.
leaky.
Thank you for starting the video.
Sorry, to potassium.
Yes.
And if it's leaky to potassium, we're going to have potassium leak out of the cell very slowly. Now, if positive potassium is leaking out of the cell, it becomes even more negative inside, right? Because positive things are going out. So it goes from negative 5 actually all the way down to around about negative 55. And so we've now got around about negative 55, a charge difference inside compared to outside. So inside is negative 55 compared to the positive outside. Now, another thing that's happening is this. These SA node cells are leaking not just to potassium, but they're also leaking to sodium and calcium. So what you'll find is...
So remember, see that he's also saying they're leaking calcium.
Is this. You're going to have some cells here. Let's draw this as a square. You're going to have some channels here, that are leaky to calcium. And I want you to think about this. Calcium sits predominantly outside the cell. And it
So higher concentration of calcium outside of the cell.
will leak into the cell. It brings a positive charge inside. In actual fact, there's also leaky sodium channels, which bring positive sodium into the cell as well.
Well, those are your funny channels. It doesn't take a lot of time to talk about that, but the other books that I have on physiology talk about that. That's what's keeping it from being a non-resting membrane potential. Your book talks about how it's non-resting because of sodium channels, but they're strange looking channels. That's why they were named funny channels. Is they're constantly leaking sodium. You didn't have these in muscle. You probably had some, but not any that were of noteworthiness.
Probably getting confused here. We've thrown 3 sodium out, 2 potassium in, but potassium is leaking out, making it negative inside, but we've also got calcium and sodium leaking inside, making it positive inside. It doesn't know what charge it is. In actual fact, what that means is this. Its baseline is around about negative 55, but because of the leaky calcium and sodium, it actually is always drifting up towards the positive, because all this calcium and sodium is slowly drifting in.
So this is what's keeping it from being non-resting. It's never stable. You never have a stable moment of the resting membrane potential. Let's not even say it, it's not resting. So you never have a stable negative membrane potential that stays consistently at one number or one voltage. It's constantly moving.
Now, in actual fact, there is a mark right here at around about negative 40. Now, this mark at negative 40 is what we term the threshold, and it's the threshold for what? Another group of channels. What are these channels? They're another type of calcium channel. At negative 40, this is the key that unlocks this new type of calcium channel, a faster calcium channel. And when that opens up, more calcium rushes in. And when more calcium rushes in, what we get is a fast stimulus up to around about positive 20. Now, when it hits, as this is happening, going up to positive 20, what you'll find is that at around about positive 20, more potassium channels open up. Remember, these were just leaky potassium channels. Now we've got very specific potassium channels opening up, and heaps of potassium start to leak out of the cell. Now, if heaps of potassium leak out of the cell at positive 20, the positive is going back out again, and it drops back down to around about negative 55 to negative 60. Now, if it's dropping back down to here, what happens again? The whole thing occurs. Leaky calcium, leaky sodium, and then it goes.
So the leaky sodium and potassium are keeping it from ever being steady, the membrane potential. Because you constantly have sodium and potassium. Once you're at a negative resting membrane potential, you start having this leaky calcium and sodium, keeping the membrane potential from ever resting. That's what's maintaining that autorhythmic. That's what kept that heart beating after I'd cut away all of the nervous system and the blood from that embryo. That's what kept it beating.
Goes back up to negative 40, which then opens the calcium channels, and that sends the signal back up, which closes the channels but opens the potassium channels, and it drops back down. So what do we have here? Basically, to summarize, we've got at this point here, it's sodium and calcium leaking in. At this point, we've got calcium shooting in. And at this point, we've got potassium going out. Now, what we term this part is depolarization. This part is repolarization. Depolarization, repolarization. And this is what's happening at both the SA and AV node. And as you can see, it just spontaneously does this. And it does it at the SA node. It does it around about 60 to 100 times per minute. And at the AV node, it does it around about 40 to 60 times per.
Remember that for one moment. The SA node will spontaneously create this rhythm 60 to 100 times per minute. The AV node is also capable of doing this from 40 to 60 times per minute. Well, why do we call the SA node the pacemaker? And why don't we just use the AV node?
Because it's faster.
You're so good. Yeah. SA node is faster. You can maintain life with the AV node. The bundle of His, or it might be the AV bundle. Yeah, the AV bundle will do it at 20 to 40. However, that's not capable of sustaining brain life of the brain. So if something happens to the SA node and the AV node, because it's the second fastest, can generate a rhythm and maintain the rhythm. But it's 40 to 60. But with the bundle of branches, it's then 20 to 40, and that's not fast enough to maintain enough blood flow going to the brain. All right, I'm gonna end the video right there. Becky, I will tell you, if your dog starts my dog barking, we're going to be in trouble.
That one's not mine. Mine's actually, my puppy's actually calmly
Okay.
laying here right now.
I don't know why I'm trying
But
this.
yes.
Okay, she hasn't started. She's now sleeping here.
Yeah, he must
I'm
have
like-
heard whoever's was barking because his ears were up. He was looking around. I was like, oh no.
Okay, come out of this. Come out, come out, come out. So impulses from the SA node are going to travel via gap junctions to the left atrium, the AV node, located in the floor of the right atrium. Then that impulse leaves the AV node and moves into the atrioventricular bundle or bundle of His that extends into the interventricular septum, where there's going to be a slight nodal delay as the impulse leaves the AV node and enters the AV bundle. This gives the ventricles just a few extra seconds, just a little, like 0.1 millisecond longer to fill up with blood. And once within the septum, the AV bundle spreads into the left and right bundles. These bundles are going to pass the impulse to the conduction cells called the Purkinje cells, or the AKA the subendocardial conducting network that begins at the apex of the heart. These fibers then spread the impulse superiorly, moving it up to from the apex to all of the ventricular myocardium. So just showing that again. Sequence of excitation. SA node to AV node to AV bundle to the Purkinje fibers up the wall. So the analogy, the reason the SA node is the pacemaker, like Becky said, is it's going to generate the fastest rhythm. I know. So what can we do? If it drops off, if something happens, the AV node will then take over. And then if it drops off, the Purkinje fibers, AKA the bundle of His, also has its ability to initiate depolarization with its autorhythmic cells. The only reason the SA node is a pacemaker is because it's the fastest. Questions? So the blank is the pacemaker that initiates each heartbeat.
C as in cat.
Yep. That the sinoatrial node. The cells of the sinoatrial node do what during the pacemaker potential?
Was it D?
D?
D as
D?
in dog. D as in dog.
Why would you say they repolarize fast?
Oh no. I think it's depolarized fast.
I would say they depolarize fast. That's the
Yeah.
closest answer here.
Yeah.
Because this answer is, this is kind of a trick question. You have to go with the most correct answer. But you know that it's not depolarized slow and then repolarized fast. Because it
Right.
depolarizes slow, then it depolarizes fast, and then it repolarizes fast. So it starts with a depolarizing fast. If the SA node is damaged, the heart will likely beat at what? What did he
C.
say when he said, what did he
It-
tell you? Go ahead.
C. I think it's C because normal would be E.
It's not C. That would be the Purkinje fibers.
Okay.
What if it's not C and it's not E?
D
D.
It's D,
as in dog. Yeah.
because that would be the AV bundle. If communication between the SA node and the AV node becomes blocked, which is most certainly affected, the ventricles will contract at a slower rate. The ventricles will not contract. The atrial will contract at a slower rate, or the Purkinje fibers will increase the number of action potentials generated.
Believe it's A.
Why would you
The
say that?
E.
Well, because if there isn't any communication, then it will definitely slow down the mechanics.
Or down the
Right.
mechanics of which one?
Which ventricle?
No, whether is it? Why would it slow down the ventricles and not the atria?
Because that's where contraction happens.
It's going to start. Well, it also happens in the atria, but
To push the blood out.
well, it also pushes. The contraction happens in the atria also. But Claire was right. The whole reason it's got to pass all the way to the AV node is to be able to initiate the ventricles to contract. But if for some reason you quit getting communication, the AV node or the bundles of His will take over. So the ventricles will still contract. They're just going to contract at a slower rate. The atria will still contract, and they're still going to contract at their normal rate because it's not stopping the current flowing in the atria. It's just stopping it between the SA node and the AV node. Does that make sense?
Yeah, it does. But I have a question. So if the SA node doesn't function like it's supposed to, does the AV node then take over, or the SA node will still function but at a slower rate?
Well, I mean, it depends on who's fastest. If the SA node is still able to function, but it's not functioning as fast, if the AV node is faster, it will take over. This idea, it will take over.
But if the SA node is still faster than the AV, even though it's not functioning, as much as it's supposed to be, between the 60 to 100, then the SA node will take over.
If it depends on whoever is faster will become the normal pacemaker.
Got you. Okay, thank you.
That's really, it's actually pretty important if you think about it, because it's what's going to stimulate the heart rate. And so that's a good clarification, Zena. Occasionally, an ectopic, if you think about like an ectopical pregnancy, something outside of the uterus, occasionally an ectopic pacemaker will develop in part of the heart's conducting system. What happens to the heart rate if an ectopic atrial pacemaker depolarizes at a rate of 120 times per minute? Well, it becomes a pacemaker, just like Zena was asking about. It becomes a pacemaker. It really is whoever's the fastest. What would increasing the potassium permeability do to the membrane potential of an autorhythmic cell? Well, what did it do in the muscle? Guys, apply this back to what you learned in the past. What would it do in the muscle if you increase the permeability of potassium? I know it was a long time ago. For you, it was probably.
Would it release, would it like increase or something about calcium?
No, it's not going to do calcium here.
No,
We're
okay.
talking about potassium, but you're probably on the right track. What did we do? What happened in the muscle if we increase the permeability to potassium? It hyperpolarized it, right? It made the resting membrane potential even lower, right? Do you remember this? Someone tell me yes.
I do remember it, but so basically it's a change in the membrane potential.
Yeah, it's going to make it much more negative. Well, how much, then what does that do with your ability to reach threshold? It goes down, right? And the result is that the whole thing is going to slow down. Does that make sense? If it's harder for you to reach threshold, then the rhythm is going to slow down. So now talked about the action potentials in the autorhythmic cells, action potentials in the cardiac contractile cells. There is a voltage gated sodium channels open. Now remember, potassium is the major player in depolarization of the autorhythmic cells. In the cardiac muscle contractile cells, it's sodium. So sodium, voltage gated sodium channels open. The sodium inflow depolarizes the membrane and triggers the opening of even more. So we get this positive feedback loop, creating and it rapidly rises the membrane voltage. At about positive 30 millivolts, the sodium channels start to close. However, at this point, calcium starts to come into the cell. And it's going to slowly help maintain, even though potassium channels are opening here too, because we're getting repolarization, calcium simultaneously is going to start entering the cell also. Remember, higher sodium inside, higher sodium outside, higher calcium outside, lower potassium outside, therefore potassium is higher inside. So higher sodium, higher calcium outside, higher potassium inside. Calcium starts to enter through through slow channels, prolonging the depolarization of the membrane. It's not, I mean, we still have potassium coming out at that point, making the membrane more negative, but the calcium is slowing it down. So rather than having a rapid repolarization like we saw before, it's going to be slowed down. Creating this plateau. And then the calcium channels start to slow down, shut off, or close out. And the potassium channels really open and allow for repolarization. So we get a rapid depolarization with sodium influx. And then potassium starts to come in. Get the potassium channels opening. However, we get calcium channels also opening, allowing for some calcium to leak into the cell while potassium is leaking out, resulting in a slower depolarization. Just kind of slows it down. And then the calcium channels close, and we get a rapid outflow of potassium. Is this clear? So the major difference here between the one that with the regards to the action potential in the neurons is that in addition, it's rather than have a rapid repolarization, calcium starts moving in at the same time that potassium is trying to move out. And the result is that we have a much slower repolarization. Resulting in what we call an absolute refractory period, where you can't get another action potential in those cardiac cells until those potassium channels close, until those calcium channels close. Sorry guys, I'm really warm for some reason. Mom, can you turn down the temp just a little bit?
It's me every day of my life. I think it's menopause. I don't know.
I think
Yours could
Definitely
actually be
menopause for me.
part of my problem is that I feel kind of brain dead this morning, and I just drank a bunch of hot chocolate, and that's making me feel warm.
Mhm.
So action potentials in the actually, bear with me here. I'm gonna do this a little differently here. So so during that repolarization, so you get the sodium moving into the cell, right? That's your rapid depolarization. And then it actually opens, and then once you get the maximum depolarization, you start to get gradual repolarization with the addition of calcium coming in to the cell as potassium is trying to leave. How does that generate contraction of the cardiac muscle? The voltage gated calcium channels open and that calcium enters the cell. The result is that calcium induces ryanodine receptors on the sarcoplasmic reticulum to open calcium. This is, at this point, this is going to work. Let's see here. Exactly like we saw with the muscle, skeletal muscle. Remember that calcium was a switch that turned on the troponin and the tropomyosin and allowed for the myosin and that to bind to the actin. So calcium induces a calcium release from the sarcoplasmic reticulum. And the calcium ions bind to the troponin, which allows for the actin to be able to, allowing the myosin to bind to the actin. Initiating the contraction. And then when the contraction is over, the calcium gets pumped back into the sarcoplasmic reticulum. And the contraction stops. You guys, if this isn't clear from you, go back and look at the way the skeletal muscle contracts, because it's happening the same way at this point. The only difference here is that we're getting a calcium signal that's initiating the calcium release from the sarcoplasmic reticulum. It's not sodium, it's calcium. I'm going to come back to this actually. Yeah, we will talk about this. So action potential in the cardiac contractile cells. That calcium potential change travels along the T tubules, causes a release of calcium from the sarcoplasmic reticulum, which ends up with an elevated amount of calcium inside the cells. The troponin and tropomyosin is shifted away from the actin binding sites for the myosin, or the myosin binding sites on the actin. You get cross-bridge cycling between the thin and thick filaments. Remember the myosin and the between the thick filaments, the myosin, and the thin filaments of the actin. The thin filaments slide inward between the thick filaments, just like we saw with the skeletal muscle, resulting in contraction.
Can I ask a question?
You can.
Is troponin a cardiac cell or it's a protein?
You know
It's
what?
not an
I'm
ion,
gonna
right?
make you, Zena, I'm gonna make you go back and look at, it is, I'm trying to think, and I've got to get through a certain amount of material today, so I'm trying to think, how do I say this, make it easy? I'm gonna send you a clip and I want you to look at it. Because what it is it's essentially, it's a signal when the myosin overlaps with the actin to allow for muscle contraction. The troponin blocks that interaction between the myosin and the actin,
Okay.
stopping contraction until you want contraction to occur. And the way that you remove the troponin from the actin is by binding it to calcium. When the calcium binds to the troponin, it shifts the troponin so that the actin binding sites are now exposed to the myosin. And that allows for muscle contraction. Does that make sense?
It does, yes.
I'm sorry, I got a little impatient there and I shouldn't be that way.
No, you're fine.
The plateau in the action potential of the cardiac muscle results from the action of what? Guys, come up, look at this. What do you think it is? This is the plateau.
Sodium.
What? No, look at, don't, Claire, look at it. What's this plateau being caused from? Read this and tell me what is causing this plateau.
Ca2, calcium,
Entering,
entering through slow Ca2 plus channels, prolongs depolarization of membrane, creating a plateau.
so now tell me what's creating it.
Calcium, D. Wait, yeah. Did it say slow or fast?
Is it really
E
small?
is in
E.
Is it
L,
really small?
slow.
Claire, is it really small?
Did it say small or slow?
Did you just, is it hard for you to read because it's small on your screen?
Well, I have it on my computer too, but
Okay.
I was looking. Yeah, I'm using my phone because my electricity's back on. Thank
Okay,
God.
good. Okay.
I was-
Well,
But-
it's slow calcium channels because right here, so calcium is entering through slow calcium channels that prolongs the depolarization of the membrane, creating this plateau. When sodium channels are fully open, the membrane of the ventricular cardiomyocyte, what? What does sodium do?
Would it be
God.
C?
It's C. Good job, because it's depolarizing, right?
Right.
And it's very fast. So just another picture of that, showing this absolute refractory period. Because of the calcium. Calcium is slowing it down, causing this absolute refractory period. If you remember when we talked about skeletal muscle in 121, you get tetanus. You can get that non-fused tetanus where muscle contraction, one muscle contraction can stimulate another muscle contraction because you don't have enough time to move all the calcium back into the sarcoplasmic reticulum before it happens again. In this case, you don't ever get that tetanus. You don't ever get unfused tetanus. The calcium gets completely pumped back into the sarcoplasmic reticulum before another contraction is initiated. Because of this slow repolarization. So this absolute refractory period is caused by the calcium slow channels doing this slow repolarization. So the calcium, oh, excuse me, Mary, really? Oh, no, that isn't the answer. The blank provides most of the calcium needed for myocardial contraction. If you don't know what this is, think about what it was for the skeletal muscle because it's the same thing. Somebody tell me, what did it store it for the with regards to the, say it again?
C.
C as in Charlie, you're right, Rachel, it's C.
And you can hear me.
Yes, I can. If tetrodotoxin, a poison that blocks sodium channels, is applied to cardiac contractile cells, what do you think is going to happen? What do you think is going to happen? You guys, what's going to happen if you block sodium coming into the cells?
Heart
Well,
attack.
the voltage. Yeah, I was gonna say the voltage gates won't open, so you're not gonna get. It won't be good.
Well, we know that. Oh, that was funny. Nope, it won't be good. You're right.
It's not gonna depolarize.
I needed that this week. That was funny. It's not going to depolarize. So you're not, it's never going to have a membrane. It's not, you're not going to initiate any action potential. Let me, I'm going to tell you something really quick, and then I won't bring it up again. I may have brought it up last semester. One thing that they do, you know, you can apply a tetrodotoxin to stop a membrane potential. If you, and my mother loves how I read a lot of violent books. I told you that I knew how blood flows because I watch NCIS. and she's in another room. So she puts, I don't, when I'm listening to something really graphic, I put on my headphones. But the way that they kill people by lethal injection is by giving them a bunch of potassium chloride. What that does, what do you think it does?
To put you in like cardiac arrest.
Well, how does it do
Like
that?
stops,
How does it do
well,
that?
it stops, or just, hold on, let me think. So it like slows everything down.
Well, how are you sure it's going to slow everything down, or is it going to stop it?
I would say stop it
And why is it going to stop it?
because it's too much potassium and
What
sodium,
does
so
that-
it's not going to
No,
depolarize. Like it,
it's just potassium. It's just potassium. Bunch of potassium chloride. Where is potassium the highest, inside or outside of the cell?
outside,
Nope, nope, Becky.
inside, inside.
Kind of putting her on the spot, and it's still early in the morning.
Yeah,
So it's higher on the inside of the cell. Well, what if we make it the same on the outside? There's no reason for the potassium to flow at that point from the inside and repolarize the cell. And the result is that the membrane potential becomes 0. Does that make sense?
yeah,
Does it, Becky?
yeah, it does.
Okay.
Because it's like equal on both sides and it's not going to do anything
Right,
in like layman's terms, you know what I mean? Like in a generic, to
right.
make it, yeah, it makes sense.
I'm sorry, you guys. I've had kind of an interesting week this week, and I just told my mom, I said, all I have to do is make it through this one today. And I apologize if I'm being a little uptight and a little impatient, and I need to stop.
You're totally fine.
Well, I still feel that way. ECGs, so electrocardiographs. Remember this from before. When in lab we had that the QRS, and the T wave. The P wave was atrial depolarization. The QRS complex is ventricular depolarization. And the T wave is ventricular repolarization.
I'm sorry, can you repeat that? So P wave is atrial.
P wave is atrial depolarization. Excuse me, yeah, atrial depolarization. The QRS wave or complex is, my mom's helping me in the background, ventricular repolarization, excuse me, depolarization. Sorry, let me start again. P wave is atrial depolarization. The QRS wave complex is ventricular depolarization. And the T wave is ventricular repolarization. Well, we know that repolarization always follows depolarization. Where does the atrial repolarization occur? It actually occurs during the QRS complex here. But because this is such a big complex, it's camouflaged within it and you don't see it. But it's going to happen then. So remember, repolarization follows you have depolarization, then you have repolarization. Depolarization, then repolarization. So the atria depolarize in the P wave. Then the signal spreads down the AV node into the bundle branches. And as it's spreading down, that's the QRS complex. It's depolarizing. During this time that the ventricles are depolarizing, the atria are repolarizing. But we don't see that because this is such a big area in comparison to this on the ECG. And then the T wave is repolarization of the ventricles. Is this clear? So what an ECG measures is it's measuring the electrical conduction through the heart. It's measuring the depolarization of the atria, the depolarization of the ventricles, and the repolarization of the ventricles. If the repolarization of the atria didn't occur at the same time that the depolarization of the ventricles is occurring, we'd see it too. But we don't because this QRS complex is so prominent. I should have put this before this, but I didn't. I'll switch around these slides later. But ECG reading versus atrial and ventricular depolarization. So here's our action potential of our ventricles. Right here. Here's the depolarization. Here's the depolarization of the ventricles. Here's that repolarization right here. Here's the atria, here's the ventricles. So here's this spike. So the ECG is measuring the action potentials through the cardiomyocytes. And we're seeing this sharp spike where with the depolarization of the atria, the P wave. Then it slows, and at the same time that we're getting the repolarization of the atria, we're getting the depolarization of the ventricles, this sharp spike, which then slowly repolarizes, which is where the T wave is. So it's really hard to measure the muscle contraction of the heart. You could do it with an MRI, but an easy, faster way that's going to take less time and be easier to measure is to measure an ECG, to initiate or estimate what's going on with the heart contractions by following the nerve impulses or the conduction. Is this clear?
What is the difference? Like when I worked at UPMC, I did a ton of EKGs, but it's basically the same.
Yep, it
Seems
is.
like
It's the same thing.
Yeah.
The K depends on where you learned how to do it and where you learned how to say it. K and C, cardio in England is spelled with a K. Cardio
Okay.
in the US is spelled with a C.
I was
Okay,
wondering
'cause-
that too, because I also learned on an EKG machine, and
Yeah,
now at the new
and
hospital,
all the
I'm learning
books called ECGs now, so we have
So I
to, mother,
wondering if they were the same thing.
what'd you say? As my mother's talking to me in the background?
I said I also learned on the EKG, and then now I'm working at a WellSpan hospital, and they do ECGs, and I was really wondering if it was the same thing, because it was exactly the same thing.
Yeah.
It's the
Machine.
same thing.
So
Yeah, it's like the printouts look the same.
exactly, yeah.
My mom was saying traditionally and they've always called them EKGs. And I said, but mother, all the textbooks call them ECGs now. So we call them ECGs.
Yeah, I was so confused.
Tomato.
You have to get
Yeah.
with the times. All right. So just another picture of this showing the spread. So this area, this PQ area right here, this is the AV node move. Remember that slow AV node moment where it's spreading from the SA node. It has to go through the AV node to start in the ventricles. That's this PQ segment. ST segment is the time it takes for the ventricles to be completely depolarized and to start to repolarize. The PR interval is how long does it take for from the time that you start the atrial depolarization to where you start the ventricles depolarizing. Another picture just essentially showing the same thing. So the P wave is atrial depolarization. The QRS complex is ventricular depolarization. The T wave is ventricular repolarization right here. The PR interval, so this area to this area, is a conduction through the AV node before activating the ventricles. If you're someone who regularly reads ECGs, long gaps between these can be the result of a heart block, a block in the conduction system. That's why these intervals are also important, and why the lengths of these and the lengths and their magnitudes are important. So signal conduction through the AV node before activating the ventricles. The QT interval is the duration in the ventricular depolarization that's shorter during exercise. QT. The QRS interval is atrial depolarization, repolarization, and diastole, heart, remember that's heart relaxation. And where the repolarization is of the atria is being concealed by the QRS complex. The PQ interval is a signal conduction from the SA node to the AV node where atrial systole begins. ST segment is the ventricular systole and the ejection of blood that corresponds to a plateau of cardiomyocyte action potentials. ST. Questions? So a normal sinus rhythm, remember that sinoatrial node, that's where we get that sinus rhythm, because this is the rhythm being initiated by the sinoatrial node. Looks like this. Don't bark, don't bark, don't bark. Sorry, someone banged the door outside of my apartment and I was just waiting for my dog to bark. Don't bark. Don't bark, don't bark. Ventricular fibrillation, where most likely it's only the bundles of hisses that are stimulating the action potential. Actually, let's not even say that. There's random action potentials being spread. But because they're so random and atomic, there's no dominant one. Atrial fibrillation, there's nothing coming from the atria, therefore the only ones you have are the signals coming, the action potentials being initiated by the bundle branches. Within the ventricles. This is an ectopic premature ventricular contraction. Oh, this is a heart block. So you see there's some P waves, but then there's no following QRS complex and a T wave. Meaning that the SA node may be firing regularly, but there's a block somewhere between the AV node and the bundle branches. Resulting in no depolarization of the ventricles and repolarization. Or it's partial. Get one, but then you don't always get the ventricles depolarizing. Then premature ectopic ventricular contractions, where you don't have a P wave. You just have a mini ventricular ectopic contraction. Questions? You know, I don't want to take a break, but I do, because I think I'm getting tired of sitting in one place, but I know how much we need to get through. But I still think we should take a break. Because if I'm tired of sitting in one place, you guys probably are too.
Yes, ma'am.
I'm not going to take very long. Let's just take 5 minutes and come back to right here. All right, come back, missus. We have to teach. Come on, Pacers. Okay. All right, so coming back to this. Atrial systole. Guys, when would it occur? If contraction follows the action potential, when does atrial systole begin?
B,
Say it one more time.
B as in boy.
You're right, Rachel. So it's going to happen during the P wave. So during the repolarization, during the depolarization, it's going to happen right in the middle of it. After a heart attack, which of the waves of an ECG are most likely to show the changes? Heart attack meaning loss of the muscle.
Is that P wave? Or no?
It's well, what starts the P wave? What is going to start? What's going to start depolarization of the atria? The SA node is going to start depolarization. So is it going to be the one affected first? Heart attack meaning the death of the muscle. It's not going to be affected first. If the SA node is right up in the very top of the atria, and the heart attack is the death of the muscle, the result is that it's actually going to be the QRS and the T waves. Because it's not going to affect that initial stimulus. Like I said before, that autorhythmicity is pretty amazing. Told you about that chick that there requires very little for that to be initiated. So it's actually going to be the QRS and the T waves, because usually what it's the ventricle muscle that starts to die with a heart attack. It's not the atria. And that the result is that you see the QRS and the T waves being the ones that are most affected, because those are the ones most affected. Those are the ones that show the activity of the ventricles. Is that clear?
Yes.
An extended period of time between the P wave and the QRS complex may indicate which of the following. So this is the difference. The P wave, remember, that's the depolarization of the atria. QRS complex is a depolarization of the ventricles. Extended time between the P and the QRS complex may indicate which of the following. Sorry.
Would it be C?
I would just say it's C.
Because usually the longer the time is between like the P wave and the QRS complex means like there's some type of like blockage or
I
slower
think
contraction.
you're right. I think you're right, Martin. Because remember, we're looking at this electrical conduction. Actually, yeah, I think you're right. Because we're looking at this electrical conduction to estimate what's going on with the contractions, because that's something we can't measure as easily. If there's a blockage between the AV bundle and the AV node and the AV bundle, how will this affect the appearance of the ECG? The blockage between the AV node and the AV bundle is the PR interval going to become slower, meaning how fast the depolarization spreads from the atria into the ventricles?
Yes.
You sure? Is it going to become fat? Is it going to, I'm sorry, it's going to become slower. So is this going to become smaller or larger, this interval?
The interval is going to be larger.
Right, so this isn't the right answer, right? A is not the right answer.
Right.
What about this
Sorry.
one? The QRS interval would be longer. Between the AV node and the AV bundle.
I would say yeah.
It's not going to be longer. And the reason is, I'm sorry, Martin, it's not going to be longer because once it's in the ventricles, it spreads fine. The QRS complex is this right here. Once it's in the bundles, it's going to spread fine. It's just not going to spread fine from right here, where the AV node is, to right here. Does that make sense? Not going to spread very well from right here. If there's a block between here, it's not going to spread very well right here. So it has to be something right here. Does that make sense? Maybe. If it's right here, here's the conduction, here's the AV node, here's the bundle. Once it's here, it's going to spread fine. But from right here to right here, it's going to slow. which means it has to be, it's going to be right here. Is that clear? Somebody say yes or no.
Yes.
It's getting there. It's a it's a tough concept, but
I understand. So what there's going to be is there's going to be more P waves. You're going to get easily atrial depolarization. You're just not going to get repolarization. Well, or you're not going to get the QRS complexes. Does that make sense? You're going to get really easily atrial depolarization. You're just not getting repolarization. Excuse me. You're getting easy atrial depolarization, but you're not getting ventricle depolarization as fast. Because of that block. This top picture ventricular fibrillation. I'm sorry, guys. I've got to keep going ahead. I realize I'm a little behind, and therefore I want to keep moving forward. Spend some time with this. Recognize that all the ECG is measuring is the conduction. If it slows down at some point here, one of these is gonna become longer, or it's not going to occur at all. If you have a block between the AV node and the AV bundle, you're still going to get depolarization of the atria. But whether or not you get depolarization of the ventricles is unpredictable. The bundle of His by itself can initiate an action potential. It's just not going to do it as often. So you're always going to get P waves, but you may only get QRS complexes, the ventricle depolarization, every once in a while. For this one, with QRS complexes, then P waves, you may always get if you get more QRS complexes than P waves, you're getting more ventricular depolarization than you are depolarization of the atria. So this is ventricular fibrillation. And so what's more devastating, atrial fibrillation or ventricular defibrillation? Well, the ventricles are the ones that are pushing the blood out into the heart and the lungs. Therefore, it's more devastating if it's ventricular. pulmonary versus systemic edema. If the blood output from the right, gotta think for a second. If there's buildup in the lungs because of the left ventricle, the part of the left heart that's not functioning effectively, fluid will accumulate in the pulmonary tissues or in the lungs. It will back up. It's going to consistently back up. So eventually it's going to affect systemic too. But right now it's just in the lungs. It's just a buildup. And if it builds up in the lungs, it's going to build up in the right part of the heart also. In systemic edema, the left ventricle is struggling. And the result is that we have fluid buildup oh, wait, I'm sorry, bear with me here. Okay, let me go back. Pulmonary edema is left ventricle failure because it's building up in the lungs. Systemic edema is right ventricular failure because it's building up in the tissues because we're not being able to pull it out of the body fast enough and move it into the lungs. Questions? So one is building up in the lungs because the left ventricle, left part of the heart is failing. Systemic edema is buildup in the tissues because the right part of the heart is failing. Questions? Guys aren't saying anything. So we're going to move on here. The valves, 4 heart valves. The tricuspid valve and the bicuspid valve, which are separating the atria from the ventricles. Then we have the aortic valve and the pulmonary valve. The tricuspid and the bicuspid valves are being held down along the walls of the ventricles by the chordae tendineae. Remember that from earlier. The aortic and the pulmonary valves are semilunar valves. They're not being held down like that. They have little cusps. So the right atrioventricular valve is located between the right atrium and the right ventricle. And it's got 3 shaped cusps of dense connective tissue covered by endothelium. And the chordae tendineae are attached to the free edges and hold the cusps down on the sides of the ventricle walls. And the right atrioventricular valve is going to function in preventing backflow of blood into the right atrium when the ventricles contract. The pulmonary semilunar valve is between the right ventricle and the pulmonary trunk. It's got 3 semilunar cusps of dense connective tissue that are covered by endothelium. And there's no chordae tendineae here. Semilunar valves, neither of them have chordae tendineae. This pulmonary semilunar valve is going to prevent backflow of blood into the right ventricle when the ventricles contract. The left atrioventricular valve is going to be between the left atrium and the left ventricles. It's got 2 triangular shaped cusps of dense connective tissue that are covered by endothelium. And the chordae tendineae are going to be attached to the free edges, holding it onto the walls of the left ventricle, preventing backflow of blood into the left atrium when the ventricles contract. The aortic semilunar valve is between the left ventricle and the ascending aorta. The 3 semilunar cusps of dense connective tissue covered by the endothelium with no chordae tendineae. Remember, the semilunar valves have no chordae tendineae. The aortic semilunar valve is going to prevent backflow of blood into the left ventricle when the ventricles contract. So those are our 4 valves. 2 atrioventricular valves, 2 semilunar valves. Showing the cusps of the atrioventricular valves being held down on the walls of the ventricles by chordae tendineae and papillary muscles. Another depiction of that. Here's our bicuspid and our tricuspid valves. Here's our aortic semilunar valve and our pulmonary semilunar valve. One is being held down by chordae, 2 are being held down by chordae tendineae. 2 are not held down by chordae tendineae. And are openings into arteries. 2 are openings between the atria and the ventricles. These valves work as mechanical devices that function in response to blood flow. They function on the principle that they are going to stay open as long as the blood pressure is greater in the emptying chamber. When the pressure in the chamber that they're emptying from, when the liquid, remember we're just looking at liquid here, when the liquid pressure closes in the empty or drops in the emptying chamber, those valves are going to close because things are always moving from a high to a low. The valves close when the pressure gets too high in the filling chamber, keeping the blood flowing one direction. So mechanical devices that are functioning in response to blood flow. When the flow is too high, they close. Preventing backflow of blood. A good analogy of this is your syringes. If you pull up Sharon, here I have a syringe. I wanted a needleless syringe, but my mother didn't have one, so we got what we have what we have. So if I increase the volume in my cavity, I decrease the pressure there, right? Decrease the pressure within the chamber of the syringe. And the result is, because things always move from a high to a low, the outer pressure here is now greater than the pressure in here. And therefore, because air always moves from a high to a low, it's going to flow within to the syringe, right? So it's going to flow into the syringe at that point. If I decrease the volume within the chamber of the syringe, the pressure is now higher inside the syringe than it is out here. And the result is that the air is going to move out of the syringe. It's all based on where the pressure is the highest. Because things always move from a high pressure to a low pressure. So depending on where the pressure is highest, things will move in or out of an area. Well, how do these valves work? They work with regards to pressure gradients. The atrioventricular valves. So remember, they're tied down on the inner walls of the ventricles. Blood returning to the heart fills the atria. Well, because blood is like any other liquid, it's going to flow down in response to gravity. So the blood returning to the heart fills the atria, and then it continues to flow down the valve into the ventricles, because the pressure is lower in the ventricles than it is in the atria once the atria start filling up with blood. So the fluid is going to flow down. Also because of gravity too. So the atrioventricular valves are forced open, and as the ventricles fill, the valves are just going to hang there limply. When the atria contract, they're going to force an additional amount of blood through the valves. Just flows just like any other liquid. When the ventricles then contract, now the pressure is higher in the ventricles than it is in the atria. And the result is that the blood is going to try to flow back into the atria. However, when it tries to flow that way, the blood is going to come against the valve cusps, and they're going to close. And the papillary muscles contract and the chordae tendineae tighten, resulting in closure of the space, the opening. Is this clear? It's the same way for any fluid moving via a valve through an area where a valve is. The valves are going to prevent backflow. So this opening and closing of valves is determined by the pressure gradients of the blood. If the pressure is higher in, excuse me, if the pressure is higher in the atria, the blood is going to flow down into the ventricles. Once the pressure becomes higher in the ventricles than it is in the atria, the blood is going to push up against these valves, closing them off. And prevent the blood from moving back, having backflow. The strings are going to prevent the cusp from being blown out and blowing out the opposite way, or prolapse. Mitral valve prolapse, which you may have heard of before, is when there is a blowout or a little bit of a blowout. When the tissue of the mitral valve leaflets or chordae are abnormally stretchy or weak, and therefore normally, like the valves they seal it off so the blood can't flow back into the atria. Well, when there's a mitral valve prolapse, when the ventricles contract, one or both of the flaps can collapse backward into the atria, letting a small amount of blood leak back into the atria, resulting in regurgitation. Very minor differences between the atrial valve function and the semilunar valve function. Semilunar valve function, the ventricles are then contracting. And as the pressure in the ventricles contracts because the volume is shrinking, the blood is pushed up against the semilunar valves, forcing them open. When the ventricles then relax, when they go into diastole and they relax, the volume within the ventricles increases, causing the pressure to drop within the ventricles. And the result is that the blood tries to flow back into the ventricles. And the result is that the blood fills up the cusps of the semilunar valves, forcing them closed. When the left ventricle contracts, here, the blank valve closes and the what valve is pushed open.
The aortic valve opens.
Oh, nope. Yep. No, nope, no. You're right. No, you're right. I'm so sorry. Yes, you're right.
And so it would have to be E. Oh, no, I'm reading it backwards. Yeah.
You're right about the aortic valve.
Yeah. So.
But what valve is
When they,
close there?
so it would have to be the pulmonary valve is pushed.
Nope. Nope. Where's the?
Oh, no, I'm reading. Okay. So the blank valve closes, the aortic valve is open. So C.
Are you sure?
No.
Right. Why? The only reason you're right up until a point, but the left ventricle is contracting. That's on the left side of the heart. So is it the right AV valve that's closing? What's the other name for the left AV valve?
The mitral valve.
Right.
D.
So it's D.
Okay.
Congestive heart failure occurs if the heart cannot contract or fill adequately. So when it can't contract systole or relax adequately, that's congestive heart failure. The result is that you're going to get possibly pulmonary and systemic edema. Shortness of breath, swollen legs because of edema in the systemic edema, shortness of breath because of pulmonary edema, and a rapid heart rate because you're not getting a decent cardiac output. Four valves during systole and diastole. During systole, which is, remember, that's contraction of the heart, the tricuspid and bicuspid valves are closed. During diastole, when the heart is relaxing, the pulmonary and aortic semilunar valves are closed. talking about the ventricular systole and diastole. When the pressure in the ventricles exceeds the atria, the blood push in the ventricles is going to push up against the bicuspid and the tricuspid valves to try to re-enter the atria, closing off those valves. At the same time that that pressure is building up in the ventricles because of systolic contraction, it's going to push open the pulmonary and aortic semilunar valves.
Thank you for explaining that, because I was looking at this picture and I really didn't know what I was looking at. So now it makes sense that you say.
Yep. You're welcome. Let me finish explaining one more part of it, and then you're welcome. During diastole, when the ventricles are filling, the pressure inside the ventricles is being, is lower than that in the pulmonary trunk and the aorta. And the result is that the blood is going to actually try to flow back from the pulmonary trunk and the aorta back into the ventricles. But it can't, because it's going to fill up these cusps and block that flow. Does that make sense too, Zena?
Mm-hmm.
Awesome.
Yep.
You guys, what I will do, we're going to go for the next 4 minutes. And what I'm going to do is we definitely are taking our hour and 20-minute break. I'm going to take a nap. And we're going to come back. We will go through the lab. Some of it is a review from last week. Some of it's not. And then I'm going to try to hit more lecture material, because we're a little behind in lecture. And I think it's good, and it's going to help, because we're going to see this same idea of blood flow. We're going to see it in the blood vessels, which is our next section. But let me talk really quickly about this, and then we'll stop here. Heart sounds, that lub-dub sounds. You've heard these. Boom, 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 boom. The first one is associated with the AV valves closing. When the blood is when the ventricles start to contract and the blood pushes up against the AV valves, forcing them to close. The second sound is associated by the semilunar valves closing at the beginning of ventricular diastole, because the ventricles start to relax and the blood that's now been pushed up into the aorta and the pulmonary trunk tries to flow back, because the pressure is now lower in the ventricles. And it can't, because it fills up those cusps and closes them. What you're hearing is turbulent blood flow against those valves. That's the first sound, is the turbulent blood flow against the AV valves. The second sound is turbulent blood flow against the semilunar valves. Is that clear? So first sound is the AV valves closing. At the very beginning of ventricular systole contraction. And the result during that time is that the pressure in the ventricles is going to exceed that in the aorta and the pulmonary trunk, and it's going to push the semilunar valves open. And then diastole is going to occur. The ventricles are going to relax. The blood is going to try to flow back. And it's going to close those valves, the semilunar valves, resulting in the second sound. We're going to skip over this one right here. I'm going to come back to right here, and then we're going to look at the cardiac cycle. And like I said, I am going to we will go through the lab, but then I want to talk a little bit about, continue with lecture material. But so we'll start right here. When we come back, we'll finish lab, we'll go through the lab material, and then we'll talk about lecture. We'll continue on with lecture. So I'll see you in an hour and 20 minutes.
All right, see you then.
See you then.
Are any of the people who are still showing their names here, are you still present? Okay, I'm going to end the meeting.